Dear fellow scholars, this is a channel with a host. Today, we are going to talk about a very interesting paper that appeared in Nature Metabolism recently. It is entitled GABAergic Neurons Mediate the Anorexiogenic Effect of a Glucose-Dependent Insulinotropic Polypeptide Receptor Antagonist. And in this episode, we are going to discuss the broader implications of this work in the context of the incretin system and its potential for treating metabolic disorders. We lines also touch upon another recent publication that uses a similar experimental approach to study the role of the GIP receptor. Okay, so what's all the fuss about? Well, there has been this narrative in the field for some time now that the glucose-dependent insulinotropic polypeptide receptor, the so-called GIP receptor, is not a relevant drug target because its activation leads to undesirable metabolic side effects. That view is nicely challenged by this elegant study, but let's start from the beginning. You see, the incretin hormones, particularly the two most studied ones, glucose-dependent insulinotropic polypeptide, GIP, and glucagon-like peptide 1, GLP-1, are produced by intestinal L cells. These hormones are released in response to nutrient ingestion and play vital roles in regulating metabolic processes, particularly energy balance and glucose homeostasis. Their effects are mediated through their respective receptors, the GIP receptor, GIPR, and the GLP-1 receptor, GLP-1R, which are found on various cell types, including pancreatic beta cells, neurons in the brain, and various metabolic tissues. Now, while GLP-1R agonism has been consistently shown to promote weight loss and improve glycemic control, the role of GIPR has been less clear. Data regarding GIPR modulation has been somewhat ambiguous. Some studies suggested that GIPR activation might actually contribute to weight gain and worsen glycemic control. Hence, the idea that GIPR might not be a good drug target. Now, this new work by 𝗗𝗘𝗖𝗢𝗓𝗏 and colleagues beautifully challenges this notion. They show that activating the GIP receptor actually leads to weight loss. So they measured the weight of mice after they were given a GIP receptor agonist. What they found was that the animals lost weight. This finding aligns well with earlier observations that had hinted at a potential role for GIPR in energy balance and metabolism. However, what makes this study exceptional is the mechanistic insight it provides. Using a combination of pharmacological, genetic, and chemogenetic approaches, they uncovered a novel pathway through which the GIP receptor influences energy balance. They demonstrated that GIPR activation, at least in the context of their experiments, does not act directly on pancreatic beta cells to modulate insulin secretion, as was previously assumed. Instead, they found that the GIP receptor's influence on weight is mediated by GABAergic neurons. GABA, if you remember from my previous episodes, is a major sleep-promoting neuroactive compound in the brain. Okay, so how did they find this out? Well, they recorded the activity of single neurons in the brains of freely moving mice, and found that the GIP receptor agonist increased the firing of these GABAergic neurons. Moreover, they showed that these neurons project to the dorsomedial and the ventromedial hypothalamic nuclei to regions crucial for feeding behavior and energy homeostasis. And when they artificially activated these very same neurons, it led to a reduction in food intake and weight. Conversely, blocking the activity of these neurons led to weight gain. What's more, they showed that the effect of the GIP receptor agonist is dependent on the neurotransmitter GABA, and that it acts upstream of the neuropeptide Y5 system, an important regulator of energy balance. Neuropeptide Y, if you remember from my previous episodes, is a potent appetite stimulant. Therefore, inhibiting this pathway would lead to reduced food intake and subsequently weight loss. Taken together, their data paints a clear picture of a novel mechanism by which the GIP receptor, at least in this experimental setting, regulates energy balance. It appears that GIPR activation leads to weight loss by enhancing the activity of specific GABAergic neurons, which then inhibit the Y5 neurons and thereby reduce food intake. So what does this mean for the field? This discovery suggests that GIPR agonism could represent a valuable therapeutic strategy for treating metabolic disorders such as obesity. Furthermore, it reinforces the notion that the brain, specifically the hypothalamus, plays a pivotal role in the incretin system's effects on metabolism and energy balance. By elucidating these mechanisms, this study not only challenges long-held beliefs about the role of GIPR, but also paves the way for future investigations into the therapeutic potential of targeting these pathways for treating metabolic disorders. By the way, another study published recently in the same journal used a clever strategy to understand the role of the GIP receptor in diet-induced obesity. They created a gut-brain conjugate by linking an anti-GIPR antibody to GLP-1 and studied its effects. This conjugate interestingly showed anti-obesity effects independently of changes in food intake, mainly by acting on circumventricular organs in the brain. These organs are essentially areas that allow for the sampling of circulating signals such as hormones and nutrients. They concluded that their conjugate likely acts on specific neuronal populations within these structures to elicit its effects. Both of these studies taken together suggest that while GLP-1 agonism has been shown to have beneficial metabolic effects, the additional modulation of GIPR, whether through agonism or even antagonism, holds promise for enhancing therapeutic outcomes. Targeting these receptors, especially in the central nervous system, offers a potential avenue for developing novel treatments for metabolic disorders like obesity. That's all for today. Thanks for watching. Subscribe and see you next time.